Welcome back to the Deep Dive. Today, we are shifting gears a little. We usually talk about these big systems, how the heart pumps, how the brain thinks. But today, we are zooming in, way in.
We're going microscopic.
Right. We're going to look at the ultimate survival machines.
Yeah.
Bacteria. And before you tune out thinking, oh, this is just high school biology all over again, let me just stop you right there.
Please do, because this is definitely not high school biology.
Yeah.
This is something else entirely.
Exactly. We aren't just memorizing organelles. We are talking about warfare. We're talking about economics. We're talking about what looks like a simple bag of chemicals that can somehow make split-second decisions about whether to eat, whether to reproduce, or even when to launch a chemical attack.
It's a fascinating way to frame it, isn't it? Because you look at a bacterium under a microscope and it just seems so simple.
Yeah, a little blob.
It's just a single cell. No brain, no eyes, no hands. But that tiny little sack of fluid is actually a ridiculously sophisticated factory. It's an economic unit.
Yeah.
It manages a budget of energy. It maintains a complex supply chain of nutrients, and it defends its territory with absolute brutality.
And for any of the medical students or healthcare professionals listening, this is the foundation for everything, right? This isn't just nice to know.
Oh, it's absolutely need to know. I mean, if you don't understand bacterial metabolism, you won't get why a patient with gangrene smells the way they do.
Okay.
You won't understand why tuberculosis loves to eat holes in the top of the lungs, but not the bottom. And if you don't understand bacterial genetics, well, you have zero chance of understanding the antibiotic resistance crisis we're all facing.
So for our roadmap today, we're using Murray's Medical Microbiology, the 9th edition, specifically Chapter 13.
That's our spine. Yep. Murray is the gold standard here. We'll also be pulling in some of the deeper mechanistic details from Jawis, where it helps explain the how and, more importantly, the why.
And what's our mission today?
It's simple, really. We want to decode the energy logic and what you could almost call the genetic social network of these bugs. That's how we're going to understand virulence and resistance.
Okay, let's start at the very beginning. The menu. We always picture bacteria on a petri dish with plenty of food. But in the human body, they're basically hunting. What do they actually need to survive?
Well, the essentials aren't that different from us, They need the building blocks of life, carbon, obviously, nitrogen for their proteins, water, of course, a bunch of ions like potassium and sodium.
The usual suspects.
The usual suspects, but there is one specific element, one particular metal, that is so crucial, it essentially triggers an all-out war between the bacterium and the human host.
I have a vague memory of learning that anemia can actually be a defense mechanism. So is it iron?
It is absolutely iron. In fact, if you take nothing else away from this whole metabolic section, just remember the iron war.
The iron war. Okay, why iron? Is it just for structural support or something?
It's all about energy. Bacteria use iron as a critical cofactor for their most important enzymes, specifically the cytochromes.
Ah, the electron transport chain.
Exactly. These are the engines that generate ATP. Without iron, the bacterial power plant just shuts down. They literally starve to death.
So the human body has to know this, right? We don't just leave precious iron floating around in the bloodstream for any bug to grab.
Exactly. Our body practices something called nutritional immunity. We lock our iron up tight. We have these proteins like transferrin in the blood and lactoferrin in secretions like tears and mucus that bind to iron with an incredibly tight grip.
So we're basically putting the food in a high-security safe and swallowing the key.
That's the perfect analogy. The bacteria are starving. The host has locked up all the food, so of course the bacteria has to have a countermove.
It has to. What is it?
They produce their own magnets. These are tiny little molecules called cytophores.
Cytophores. Okay.
Yep. Bacteria just secrete these things out into the environment. And the key is, cytophores have an even higher affinity for iron than our own proteins do.
Higher, wow.
So they float out, they find our transferrin, and they just rip the iron right off it and bring it back home to the bacterium. It's basically chemical theft on a molecular level.
That is incredibly aggressive, just stealing it.
Oh, it gets worse. Some bacteria don't just steal from the transport proteins. They decide to break into the bank itself. You know how some bacteria like Staph aureus or Strep pyogenes are described as hemolytic?
Yeah, that's when they burst the red blood cells on a blood agar plate. I always thought that was just a neat way to identify them in the lab.
It is useful for identification, but you have to ask why they do it. What's inside a red blood cell?
Hemoglobin?
And what's at the center of hemoglobin?
Iron.
Tons of it. Red blood cells are basically just little bags of iron-packed hemoglobin. The bacteria are literally blowing up the cells to loot the iron inside.
So hemolysis isn't just some random act of destruction. It's a calculated heist.
Precisely. It's a strategic move to get the one resource they desperately need to grow and cause infection.
That totally changes how I look at a blood culture plate. Okay, so that's the battle for nutrients. Let's talk about the environment, specifically oxygen.
This is a huge concept. And clinically, I think classifying bacteria by how they handle oxygen is probably the single most useful mental model you can have.
Why?
Because it tells you where in the body the infection is likely to be.
Right, because different parts of the body have completely different oxygen levels.
Exactly. So broadly, we can put them into three buckets. First, you have the obligate aerobes. These guys are just like us. They absolutely require oxygen to survive. They use it to generate energy, and if you take it away, they die.
And a classic example would be
Mycobacterium tuberculosis.
TB. That makes perfect sense. That's why you see those classic cavities in the apex of the lungs, right?
Yeah.
The very top.
Exactly. The apex of the lung is the most well-ventilated, highest oxygen tissue in the entire body. It's high oxygen real estate. So, of course, TB settles there because it's an aerobe. It thrives where the air is.
Okay, that's bucket one. What's bucket number two?
The obligate anaerobes. Now, these are the fascinating ones. For them, oxygen isn't just useless, it's poison. It is acutely toxic. If you expose them to room air, they die pretty quickly.
But why? I mean, oxygen is life for almost everything else. Why does it kill them?
It all comes down to chemistry. When you metabolize oxygen, you inevitably create these really dangerous byproducts called free radicals. Things like superoxide radicals and hydrogen peroxide.
The stuff people use antioxidants for.
The very same. They're incredibly reactive. They just go around tearing up DNA, shredding proteins, destroying cell membranes.
So why don't we die from breathing then?
Because we have a cleanup crew. We possess enzymes, specifically superoxide dismutase, or SOD, and catalase. These enzymes find those radicals and immediately neutralize them, turning them into harmless water and oxygen.
And I'm guessing the anaerobes don't have these enzymes.
They usually lack both, or they have incredibly low levels. So when they're exposed to oxygen, those radicals just build up and up and up, and they essentially rust from the inside out.
That's a very vivid image. So clinically, where do we find these guys?
In the deep, dark, oxygen-poor places. The gut is a huge reservoir, deep abscesses, necrotic tissue where the blood supply and therefore the oxygen supply has been cut off. You have to think about Clostridium perfringens.
That's the bug that causes gas gangrene.
Right. It thrives in dead, crushed muscle tissue where there's no oxygen.
Okay, so that brings us to the third group, the ones that seem to have the best of both worlds.
The facultative anaerobes. And this group covers a lot of the heavy hitters like E. coli and Staph aureus. They are the switch hitters of the microbial world.
Meaning?
They'd prefer to use oxygen because it's way more efficient, but if the oxygen runs out, they don't die. They just shrug, flip a switch, and change their whole metabolism.
Okay, let's dig into that efficiency part you mentioned. You said they prefer oxygen. Why? What's the actual payoff?
It's all about the currency, ATP. That's the energy coin of the cell.
So walk us through the numbers. How big is the difference?
Okay, let's start with the universal first step, glycolysis. This happens in the cytoplasm of pretty much every living thing. You take one molecule of glucose, you break it in half, and you get 2 molecules of pyruvate. This happens whether there's oxygen or not.
And what's the payout from just that step?
The net gain, 2 ATP.
2. That really doesn't sound like a lot.
It's pennies. It is barely enough to keep the lights on. Now, if you are an anaerobe or a facultative bug that finds itself without oxygen, you basically stop there. You have to enter fermentation.
Fermentation. This is what yeast does to make beer and bread, right?
Exactly the same concept. And here's the key thing that trips people up. The goal of fermentation isn't actually to make more energy. You're stuck with those 2 ATP you made from glycolysis.
So what's the point then?
The point is just to recycle the chemical carriers, the NADH molecules, back to NAD plus HSR. You have to do that to keep glycolysis running. Fermentation is just a survival mode to keep those pennies trickling in.
Okay, so it's a desperate measure.
It is. But here is the clinical gold. The waste products of fermentation are really messy and diverse. Depending on the bug, they just dump out different kinds of garbage.
And we can smell that garbage.
Yeah.
Or test for it.
We can do both. We can smell it and we can definitely test for it. So if the bug dumps out a lot of lactic acid, it's likely a streptococcus or a lactobacillus. If it dumps out butyric acid and a ton of gas.
Let me guess. That's the Clostridium again?
That's the Clostridium. That gas is what causes crepitus, that horrible crackling feeling you can feel under the skin in a patient with gas gangrene. It's literally pockets of metabolic exhaust gas trapped in their tissue.
That is horrifying and diagnostically helpful at the same time. What about mixed acids?
That's usually the signature of the enterics, like E. coli or Salmonella. In the lab, we use all these different byproducts to build a metabolic fingerprint and identify exactly what bug we're dealing with.
Okay, so fermentation is low energy, messy exhaust, just to stay alive. What happens if oxygen is present?
Then you get to turn on the nuclear power plant. Aerobic respiration, you take that pyruvate from glycolysis, and instead of fermenting it, you feed it into the TCA cycle, the Krebs cycle, and then into the electron transport chain.
And what's the payout there?
Instead of a measly 2 ATP, you get roughly 38 ATP.
Wait, 2 versus 38.
38. It's about a 19-fold increase in efficiency. This is exactly why facultative bacteria grow so much faster and more robustly when oxygen is available. They are absolutely flush with cash. They can afford to build new cell walls, replicate their DNA, and just divide constantly.
That math really puts it all into perspective. And the TCA cycle. I just remember memorizing that horrible circle of pain in biochem. Does it do anything else besides just burning fuel?
Yes, and this is a key term you should know, amphibolic.
Amphibolic. Sounds a bit like ambidextrous.
Very similar idea. It means a cycle can go both ways. Yes, it burns things down for energy. That's catabolism. But it also provides the Lego blocks to build things up. That's anabolism.
How so?
The intermediates in the cycle can be siphoned off. They become the precursors for amino acids, for lipids, for nucleotides. So it's not just a furnace, it's also a construction yard.
Got it. So we've covered the energy, we've covered the raw materials, now we need the instructions. Let's talk about the blueprint, the bacterial genome.
This is where the differences between us and bacteria really start to matter for medicine.
So what's the single biggest difference?
Bacteria are haploid.
Meaning they only have one copy of their chromosome.
Right. We humans are diploid. We have 2 copies of almost every gene, one from mom, one from dad. If one of our copies is broken, the other one usually takes over. We have a backup system.
And bacteria don't.
No backup. They are flying without a net. And the consequence is huge. If a mutation happens in a bacterium, it is expressed immediately. There is no such thing as a recessive gene hiding in the background.
That sounds incredibly risky for them, but I guess it also allows them to adapt at a crazy speed.
That's the trade-off. Evolution happens in real time in the bacterial world because every single change is immediately tested against the environment, for better or for worse.
Let's talk about the physical structure of that DNA.
It's usually a single circular chromosome. It's all bunched up in the middle of the cell in a region called the nucleoid. And unlike us, they don't have histones. Those are the protein spools we wrap our DNA around to keep it organized.
What do they use instead?
They use things like polyamines to help condense it and keep it from becoming a tangled mess.
Okay, now replication, the copy machine. This is going to be a massive target for antibiotics, isn't it?
Oh, it's huge. One of the best. If you can stop a bacterium from copying its DNA, it can't divide. The infection grinds to a halt.
So walk us through the machinery. I know we have helicase to unwind the DNA.
Right, helicase unzips the 2 strands, but imagine you're trying to unzip a rope that's already twisted. What happens to the rope ahead of the zipper?
It gets tighter and tighter and all knotted up.
Exactly, that's called supercoiling. If you don't relieve that tension, the DNA will physically snap under the strain. So bacteria have this amazing enzyme called DNA gyrase, which is a type of topoisomerase.
And what does it do?
It acts like a swivel. It makes a little cut in the DNA, lets it untwist to relieve the tension, and then reconnects it. It's constantly working ahead of the helicase.
And this is where the drugs come in.
This is exactly where they come in. The fluoroquinolones. Drugs like ciprofloxacin, levofloxacin, they specifically bind to that DNA gyrase and just jam it.
So the DNA just stays twisted and replication stops.
It's actually worse than that. The drug lets the enzyme make the cut, but it prevents it from reconnecting the DNA.
Oh, yeah.
So you end up with a ton of double-strand breaks. The chromosome just fragments. It's lethal.
That is a brutally effective mechanism, snapping the DNA.
It is. But speaking of speed, there's a really cool puzzle here. You know how fast a bug like E. coli can divide under ideal conditions?
I've heard the number. Something like every 20 minutes.
Exactly, every 20 minutes. But here's the catch. If you actually time how long it takes the DNA polymerase to copy the entire E. coli chromosome from start to finish, it takes about 40 minutes.
Wait a minute. The math doesn't work. How can you divide every 20 minutes if it takes 40 minutes just to copy the instructions? You'd be handing out incomplete books.
This baffled scientists for a long time. The answer is a brilliant strategy called nested bubbles of replication.
Nested bubbles. What does that mean?
Imagine you're printing a long book and it takes 40 minutes to print the whole thing. But you don't wait for the first copy to be completely finished before you start printing the next one.
You start printing the second copy while the first one is still only halfway through the machine.
Exactly. A bacterium will start a new round of DNA replication at the origin site before the first round has even finished. So by the time the cell finally divides into two daughter cells, those new daughter cells already have their own DNA replication halfway finished.
So they are. They're born pregnant.
In a genetic sense, yes. They're born with the next generation's blueprint already baking in the oven. That is how they outpace the math and achieve that mind-boggling exponential growth.
That is terrifyingly efficient. And speaking of growth, we should probably visualize that growth curve. It's a staple on every microbiology exam I've ever seen.
It is the classic four phases. You've got the lag phase, the log phase, the stationary phase, and the death phase.
Okay, let's break them down. Lag phase.
This is when you first introduce the bacteria into a new environment, a test tube, or, a patient's bloodstream. The line on the graph is flat. They aren't dividing yet.
So it looks like nothing is happening.
On the outside, yes, but on the inside, everything is happening. They're tooling up, they're sensing the sugar levels, they're checking the temperature, and they're synthesizing all the enzymes they're going to need. They're building the factories before they turn on the assembly lines.
Got it. Then comes the log phase.
Or the exponential phase. This is that classic straight line shooting upwards on a log scale graph, constant doubling. 1 becomes 2, 2 becomes 4, 4 becomes 8. This is where that nested bubble replication is running at full tilt.
Is this when they're most dangerous to the patient?
In terms of sheer numbers, absolutely. But from a clinical standpoint, this is also when they are most vulnerable to certain types of drugs.
Oh, which ones?
The beta-lactams. So your penicillins and cephalosporins. These drugs work by targeting cell wall construction, and you can only knock down a wall while it's actively being built.
That's a great clinical pearl. If the bacteria aren't actively growing and building new walls, penicillin won't work very well.
Exactly. So then, after the party at the log phase, we hit the stationary phase.
The party's over.
The party is over, the resources start to run out, waste toxins build up in the environment, the growth rate slows down until the number of new cells being born equals the number of cells dying. The population just plateaus.
And I've heard this is when things can get a bit weird for the bacteria.
This is a major stress state, and this is when spore-forming bacteria like your Bacillus or Clostridium species realize the ship is sinking. They decide to form spores.
The ultimate escape pod.
Pretty much. They pack their DNA into a tough, metabolically dormant little shell and just wait it out.
And finally, that leads to the death phase.
The decline. The toxins become overwhelming, the nutrients are gone, and the bacteria start dying off faster than they can reproduce.
Okay, so we've sorted out the energy and the replication. Now we need to actually translate that DNA code into the worker proteins that do all the jobs. Let's talk transcription and translation.
The central dogma. DNA, RNA, RNA to protein. In our cells, we keep our DNA locked away in a vault, the nucleus. Transcription happens inside that vault. Then we process the mRNA and ship it out to the cytoplasm to be read by the ribosomes.
It's a very organized two-step process. But bacteria don't have a nucleus.
Right, they live in a studio apartment. Everything, the DNA, the RNA polymerase, the ribosomes, it's all just floating around in the same room.
Does that mean, can they do both steps at the same time?
Yes, and this is a profoundly important concept. It's called coupled transcription and translation. Even while the RNA polymerase is still chugging along the DNA, printing out the mRNA strip, a ribosome, or even a whole convoy of ribosomes, will jump onto the trailing end and start making protein immediately.
Wow. That's unbelievably fast. It's like reading the ticker tape while it's still coming out of the machine.
It is. It allows for incredibly rapid response to any environmental changes. If a new sugar source appears, they can have the digestive enzymes ready in seconds, not minutes.
Let's look at the ribosome itself. This is another major drug target.
A huge one. And it works because bacterial ribosomes are structurally different from ours. They are 70S ribosomes, which are made of a 30S and a 50S subunit. Human cells have larger 80S ribosomes.
And that slight difference in size and shape is enough to keep our own cells safe from these antibiotics?
Yes. It's the principle of selective toxicity. It means we can design drugs that jam the gears of the 70S factory but just bounce right off our 80S factory.
I always struggle to remember which drugs hit which subunits. They're a good trick for that.
There's a classic mnemonic that works really well. It's buy at 30, sell at 50.
Buy 30, sell at 50. Okay, walk me through.
So AT stands for aminoglycosides and tetracyclines. They target the 30S subunit.
AT30, got it.
And cell stands for chloramphenicol, erythromycin, which stands in for all the macrolides linezolid, and clindamycin. Those all target the 50S subunit.
Okay, that actually helps a lot. Buy a 30, sell at 50. Now, there's one more weird little detail about bacterial translation that's clinically important. How do they start the protein chain?
So in our cells, the very first amino acid in any protein is always methionine. In bacteria, it's a slightly modified version called formal methionine, or FMET.
Why does that one little chemical tag matter so much?
It matters because our immune system uses it as a homing beacon. Human cells never, ever make proteins that start with FMET. So if your white blood cells, specifically your neutrophils, sniff out FMET in your tissues, they know instantly this is not self, this is an invader.
So FMET is like a chemical siren going off saying bacteria are here.
It's a powerful chemotactic agent. It literally draws the immune system right to the site of the infection. It's a major red flag for our bodies.
That's amazing. Okay, so we've got the factory running. But a good factory needs good management. It can't just run at 100% all the time.
Yeah.
This brings us to gene regulation.
The golden rule of the bacterial economy is incredibly simple. Bacteria are cheap, they're absolute misers, they will not spend a single ATP unless they absolutely have to.
So they're constantly turning genes on and off based on what they need at that exact moment.
Right. And to do this efficiently, they organize their genes into functional clusters called operons.
And the most famous one, the one everyone dreads in biochemistry, is the lac operon.
Everyone does, but let's try to simplify it. At its heart, it's just a logic gate. The lac operon contains all the genes needed to digest lactose, which is milk sugar. But you have to remember, the preferred food for E. coli is glucose. Glucose is like easy cash. Lactose is like a check that you have to take to the bank to cash. It takes more effort.
So the bacterium only wants to turn on the lactose digesting genes if two conditions are met.
Exactly. Condition 1, glucose is gone. I'm broke and hungry. Condition 2, lactose is present. There's a check on the table I could cash. It needs both to be true.
So how does the cell know this? How does it check both boxes?
Think of it like a security system with two different locks. The first lock is a protein called the repressor. This protein physically sits on the DNA, right on the operator site, and acts like a parking brake. It blocks the RNA polymerase.
And what releases that brake?
Lactose itself, well, technically a metabolite of it, allolactose. If lactose is floating around, it binds to the repressor protein and changes its shape, making it fall off the DNA. Brake release.
Okay, so if I drink a big glass of milk, the lactose gets in, the brake is off.
The brake is off, but that's not enough to get things going. You also need to step on the gas pedal. And that's where the glucose sensor comes in.
How does that work?
It's an inverse relationship. When glucose levels in the cell drop, a stress signal molecule called CAMP rises. This CAMP then binds to a helper protein called SECP. Together, the CAMP-CCAP complex binds to the DNA near the promoter and basically shoves the RNA polymerase forward, turbocharging it. That's your gas pedal.
Okay, so let's play out the scenario. I'm an E. coli. There's high glucose and high lactose available. What happens?
Okay, high glucose means your CAMP levels are very low, so no gas pedal. Even though the lactose is there and has removed the brake, the engine is just barely turning over. You get a tiny, tiny bit of expression, but basically nothing. The cell ignores the lactose because it's happily munching on the easy glucose.
Right, but what happens if the glucose runs out?
Glucose drops, CAMP skyrockets, the CAP protein binds, and the gas pedal gets stomped to the floor. Lactose is present, so the brake is already off. Boom, you get massive transcription. The factory turns on at maximum speed.
That is actually a beautiful piece of biological logic.
It really is. It's a perfect decision-making circuit built out of just a few proteins.
Now let's contrast that with the tropoperon, the one for making tryptophan.
Here, the logic is completely reversed. Tryptophan is an amino acid, a building block for proteins. Making it from scratch is incredibly expensive in terms of energy. So if you already have tryptophan available from the environment, you want to turn the factory off.
So this is a repressible system, not an inducible one.
Exactly. In this case, tryptophan itself acts as a co-repressor. It binds to the repressor protein and helps it lock onto the DNA to stop production.
But with the tropoperon, there's a second layer of control, isn't there? Something called attenuation.
Yes, this is the hair trigger. It's an even finer level of control. And it's a mechanism that is only possible because of that coupled transcription and translation we talked about earlier.
Okay, you have to explain this. This one is tricky.
Okay, visualize the mRNA as it's being printed off the DNA template. The very first part of this mRNA, before you even get to the real genes, is a short leader sequence. And this leader sequence is a test track. It has several tryptophan codons right in a row.
So the ribosome immediately jumps on and starts reading this test track.
Right. Now, scenario one. There is plenty of tryptophan in the cell. The ribosome has all the tryptophan tRNA it needs, so it zooms right through that test track. No problem.
It moves fast.
Because it moves so fast, it catches up to the RNA polymerase that's still printing the mRNA. This close proximity causes the mRNA to fold up into a specific hairpin shape, a stem loop, that acts like a stop sign. It physically knocks the polymerase off the DNA. Transcription terminates prematurely.
It's like the assembly line workers shouting back to the office, "Hey, we have enough of this part. Stop writing the manual."
That's a perfect analogy. But now, scenario two. Tryptophan is scarce.
The ribosome gets to the test track and stalls.
It stalls right on those tryptophan codons. It's sitting there waiting, where's the tryptophan tRNA? Now, while it's stalled, the mRNA that's being synthesized ahead of it has time to fold into a different shape. This alternative hairpin loop says, keep going. It's an anti-termination signal.
So the speed of the ribosome itself acts as a sensor that tells the genome whether to keep working or just give up.
It's a sensor built directly into the assembly line. It is incredibly elegant and efficient.
Before we leave regulation, we have to talk about how bacteria talk to each other. Quorum sensing.
This is pure mob mentality. Individual bacteria are constantly secreting these small chemical signal molecules, things like N-acyl homoserine lactones.
So if a bacterium is all alone,
The signal just diffuses away into the environment. Nothing happens. But if you get a billion bacteria all packed into a small space, like, say, in a patient's lung or on a catheter, that signal starts to build up. It hits a threshold concentration.
and when it hits that threshold,
They all react simultaneously. It's like a switch flips in the entire population at once, and they turn on a specific set of genes as a coordinated group.
click on it, genes.
Virulence factors, usually. Toxins. Or a big one is biofilm production.
This is the cystic fibrosis story, isn't it?
It's a tragic, classic example. Pseudomonas aeruginosa can colonize the lungs of a patient with CF. For a long time, it just sits there, relatively quiet. But it's dividing, it's counting. And once the numbers get high enough, the quorum signal hits the threshold and snap. They all turn on the genes to produce this thick, slimy alginate biofilm.
Which acts like a shield, protecting them from antibiotics and the patient's immune cells.
Exactly. If one bacterium tried to do it too early, the immune system would just wipe it out. So they wait until they have an army, and then they all fortify the castle at the same time.
It's strategic coordinated warfare. Okay, let's move to section 5. What happens when the blueprint itself gets corrupted? Mutation.
Mutation is the raw engine of evolution. You have your standard typos. A silent mutation. A base changes, but because the genetic code has some redundancy, the wobble hypothesis, the amino acid it codes for, stays the same. So, no harm done.
Okay.
A missense mutation. You swap one amino acid for another. Maybe it's a big deal, maybe it's not. Depends on where it is in the protein. Then you have a nonsense mutation. This one is almost always bad. You accidentally change an amino acid codon into a SASOP codon in the middle of a gene. The protein gets chopped in half.
And what's considered the worst one?
The worst one is almost always a frame shift. This is a disaster. If you insert or delete a number of bases that isn't divisible by 3, you shift the entire reading frame. Everything downstream from that point becomes complete gibberish. It's like deleting the space between 2 words in a long sentence. The rest of it becomes unreadable.
But bacteria have a way to deal with massive catastrophic DNA damage, right? The SOS response.
This is the Hail Mary pass. Let's say a bacterium gets hit with a heavy dose of UV light. Its DNA is shredded. It can't replicate. It's going to die.
So what does it do?
It activates the SOS system. And what that does is it switches out its normal, high-fidelity, careful DNA polymerases for these special sloppy polymerases.
Sloppy ones.
Yeah, these enzymes don't bother to proofread. They're designed to replicate right over damaged DNA. They just grab random bases and shove them into the gaps to try and bridge the breaks and finish replication.
So they are actively choosing survival over accuracy.
Yes. The mutation rate skyrockets. Most of the daughter cells will be non-functional monsters and will die, but 1 or 2 might just have a lucky mutation that allows them to survive whatever was trying to kill them. It's a desperate last-ditch gamble.
Which brings us to what might be the scariest part of bacterial genetics. the swap meet. Horizontal gene transfer, HTT.
This is it. This is why we are losing the war on antibiotics. Bacteria don't just pass genes down to their kids, which is vertical transfer. They pass genes to their friends, their neighbors, even their enemies. That's horizontal transfer.
There are three main mechanisms we need to know for the exams. Let's start with mechanism 1, transformation.
I call this one dumpster diving. A bacterium dies and it lyses, spilling its guts out. Its DNA gets chopped up and is now just floating around naked in the environment. Another bacterium comes along, sees this free DNA, and just sucks it up and incorporates it into its own genome.
But not all bacteria can do this, can they?
No, you have to be in a specific physiological state called competence.
And there's mnemonic for the ones that are naturally competent.
Yes, the one to remember is SHINE IN. That's Streptococcus pneumonia, Haemophilus influenza, and Neisseria. These guys are the professional dumpster divers of the bacterial world. This is actually how we first discovered that DNA was the genetic material through Griffith's famous experiment with rough and smooth pneumococcus.
Right. Okay. Mechanism 2, conjugation.
This is bacterial sex. This is the heavy lifter for gene transfer. It involves direct cell-to-cell contact.
And it starts with a special piece of DNA, a plasmid, right?
Correct. The F plasmid, which stands for fertility factor. A bacterium that has this plasmid is called F+, and it's the donor. This plasmid has the genes to build a physical tube called a sex palus.
It's like it casts out a grappling hook.
That's a great way to think about it. It hooks onto a nearby recipient cell, an F- cell, reels it in, and opens a channel between them. Then it copies its F plasmid and shoots the copy across the tube.
So now the recipient cell has a plasmid too.
Right. The F- cell becomes an F+ cell. And here is the terrifying part. That F plasmid is often loaded with antibiotic resistance genes. So resistance can sweep through a bacterial population like a zombie infection. Everyone it touches joins the horde.
What about HFR? That always sounded more complicated.
HFR stands for high frequency of recombination. This happens when, for whatever reason, the F plasmid gets bored of just floating around and it integrates itself into the main bacterial chromosome.
So it becomes a permanent part of the master blueprint.
Exactly. Now, when this HSR bacterium tries to mate, it starts to transfer what it thinks is the F plasmid, but because it's attached, it starts dragging the entire chromosome along with it.
It tries to move the whole library through that tiny tube.
Yes. The tube is fragile, and it usually breaks before the whole chromosome gets across. But the recipient still gets a huge chunk of new chromosomal DNA from the donor. This is a major way that bacteria can shuffle their core genetics.
Okay, that's 2. Mechanism 3, transduction.
This is the accidental tourist. This whole process relies on a mistake made by a virus.
A bacteriophage.
Right. A phage, which is a virus that infects bacteria, lands on a cell and injects its DNA. It hijacks the cell's machinery to make more viruses.
Let's do generalized transduction first.
Okay, this happens with lytic phages, the ones that just replicate and boost the cell. The virus first chops up the bacterial chromosome into little pieces. Then, when it's assembling all the new virus particles, sometimes it gets confused. It accidentally grabs a random piece of bacterial DNA and stuffs it into a new virus head instead of its own viral DNA.
So the new virus is essentially a syringe full of random bacterial genes.
Precisely. It then goes and injects that bacterial DNA into the next bacterium it infects. It's completely random, hence generalized.
And specialized transduction.
This one is much more sneaky. This happens with lysogenic phages, like lambdaphage. These guys integrate their DNA into the bacterial chromosome at a specific spot and just go dormant. They sit there, sometimes for generations, just getting copied along with the bacterial DNA.
Just waiting.
And then when they decide to wake up and leave, usually because the cell is under stress, they have to cut themselves out of the chromosome. But sometimes they cut sloppily. They accidentally take a neighboring piece of bacterial DNA with them when they leave.
And this is critically important for toxins, right?
Oh, massively important. Many of the worst bacterial toxins we know of are actually viral passengers. The mnemonic to remember for exams is ABCD.
Okay, break it down.
A is for the Shigo-like toxin, which you find in E. coli O157. B is for botulinum toxin. C is for cholera toxin. And D is for diphtheria toxin.
Wait, hold on. So the bacterium that causes diphtheria, it isn't actually dangerous on its own.
Corynebacterium diphtheria is relatively harmless unless it has been infected by a specific beta-phage. The virus literally brings the weapon to the bacterium.
That is just wild. The bacterium has a pet virus that gives it superpowers.
Okay, finally, before we put it all together, let's talk about transposons.
The jumping genes. These are discrete segments of DNA that have the ability to cut themselves out and paste themselves somewhere else, all within the same cell. They could jump from a plasmid to the chromosome, or from the chromosome to a plasmid. They're mobile.
And when you combine all of these mechanisms, conjugation, transposons, you can get the absolute nightmare scenarios, like VRSA.
VRSA, vancomycin-resistant Staphylococcus aureus. This is the perfect genetic storm, the ultimate superbug.
So walk us through how it happened. It's a horror story in 3 acts.
Act 1. Somewhere in a patient's gut, you have an Enterococcus bacterium that is VRE, vancomycin-resistant Enterococcus. That resistance gene sits on a transposon, which is on a plasmid.
Okay, Act 2. The patient has a wound infection with MRSA methicillin-resistant Staph aureus. The VRE from the gut meets the MRSA in the wound. They touch. Conjugation happens. The VRE sends its resistance plasmid over to the Staph aureus.
So now the staph has the vanc resistance plasmid.
Right, but it's not stable. Act 3 is the key. The jumping gene, the transposon carrying the vancomycin resistance, jumps from that temporary Enterococcus plasmid onto one of the MRSA's own stable plasmids, or even its chromosome. It locks in. And now you have a Staph aureus that is resistant to both methicillin and vancomycin.
And we have almost nothing left in the arsenal to treat that.
Correct. That is the devastating power of horizontal gene transfer in a nutshell. It turns individual resistance problems into systemic global crises.
We should probably mention briefly that we humans have now learned to hack this entire system. Genetic engineering.
We've basically stolen all of our best tools directly from bacteria. Restriction enzymes. Those are just bacterial molecular scissors. They evolved to chop up attacking viruses. We use them to cut and paste DNA for cloning.
And CRISPR, which is everywhere now.
CRISPR is just a bacterial immune system. It's a genetic memory bank of past viral attacks that lets them find and destroy viral DNA. We just repurposed it to edit genes and everything from wheat to potentially humans. We're really just standing on the shoulders of bacterial giants.
So let's try to wrap all this up. We've covered a lot of ground. Bacterial metabolism isn't just a list of chemicals. It's a collection of survival strategies.
Exactly. It determines where they can live, like TB, in the oxygen-rich lung apex. And it determines how we find them by the smell and chemical signature of their fermentation products.
And their genetics is just built for speed and adaptability. Haploid genomes for instant evolution, operons for hyper-efficient regulation, and horizontal gene transfer to share resistance weapons like their trading cards.
It's a dynamic, terrifying, and in its own way, a very beautiful system.
All right, it's time to test your attention. We have 5 rapid-fire questions for you. I'll ask the question, give you a moment of silence to think, and then our expert will break it down. Ready?
Let's do it.
Question 1. A microbiologist is comparing 2 cultures of E. coli. Culture A is growing in an oxygen-rich incubator. Culture B is in a sealed anaerobic jar. In terms of ATP yield per glucose molecule, approximately how many times more efficient is culture A compared to culture B?
Pause. The answer is roughly 19 times more efficient. Culture B, the anaerobic one, is relying on glycolysis followed by fermentation, which yields a net of just 2 ATP per glucose. Culture A is using aerobic respiration glycolysis, the TCA cycle, and oxidative phosphorylation yielding about 38 ATP, so 38 divided by 2 is 19. This massive energy difference is what explains the much faster growth rate in aerobic conditions.
Question 2. You are treating a patient with a severe kidney infection using ciprofloxacin. This drug works by inhibiting a specific bacterial enzyme involved in DNA replication. What is the enzyme, and what is the physical result of inhibiting it? Pause.
The enzyme is DNA gyrase, which is a type of telomerase to second. The result of inhibiting it is that the bacteria cannot relieve the positive supercoiling tension that builds up ahead of the replication fork. This tension becomes so great that the DNA physically snaps, causing lethal double-strand breaks and cell death.
Question 3. Let's go back to the lac operon. If an E. coli cell finds itself in an environment with high glucose A and D high lactose, what will the level of gene expression from the operon be? High, low, or completely off? Pause.
The answer is low, or what we'd call basal level. It's not completely off because the presence of lactose removes the repressor protein, which is the brake. But it's not high because the high levels of glucose mean that CAMP levels are low. Without high CAFP, the CIP protein cannot bind and act as the gas pedal. The cell will always prefer to use the easy energy from glucose first.
Question 4. A non-encapsulated strain of Streptococcus pneumonia acquires the ability to make a capsule by picking up free DNA from a least encapsulated neighbor in its environment. What is this mechanism of gene transfer called? And what term describes the physiological state required for the recipient bacterium to do this? Pause.
The mechanism is transformation. The physiological state is called competence. And remember the mnemonic for naturally competent organisms that are important clinically: Shin streptococcus pneumonia, Haemophilus influenza, and Neisseria.
Excellent. Final question. Question 5. You're investigating an outbreak of Shigella dysentery. You find that the gene for the shigatoxin is located on the bacterial chromosome right next to the integration site for a bacteriophage. It appears the gene was brought into the cell by a virus that integrated and then later excised imperfectly. Is this an example of generalized or specialized transduction?
This is a classic example of specialized transduction. The key clues are the involvement of a lysogenic or temperate phage that integrates into the DNA at a specific site, and the fact that it's a specific gene, the one right next door, that gets transferred. Generalized transduction is a random packaging error during a lytic cycle. Specialized is a specific excision error from a lysogenic cycle.
So how did you do? If any of those were tricky, go back and review Murray Chapter 13. Pay special attention to the diagram of the Lycopron logic gates and really trace the lines of the 3 different HGT mechanisms until they're clear.
And maybe one final thought to leave you with. We talked a lot about speed and efficiency today, E. coli dividing every 20 minutes. But scientists have also found bacteria living in deep ocean sediment millions of years old that are alive but divide maybe once every 1,000 years. Their metabolism is barely a whisper. So how does a regulatory system like attenuation, which depends on the speed of a ribosome, even work on that timescale? The fundamental rules of metabolism might be universal, but the element of time can change everything we think we know.
Something to keep you up at night. Thanks for diving deep with us. We'll see you on the next one.
Stay curious!